Hi everyone. We will start with the first chapter of botany, that is bacteria. So what are these bacteria? So we all know that these bacteria, they are unicellular. Okay, they are unicellular microorganisms and they range in length from few micrometer. Their length is few micrometer to a half micrometer. To half a micrometer. Okay, to half a micrometer. So these bacteria, they are a variety of shapes. They can be cocci, they can be bacilli, or they can be spirally. And these bacteria, where they are found in every ecosystem. They are found in every ecosystem, and every ecosystem on earth, and they are around, we can say they are around 5 into 10 to the power 30 bacteria on earth. Can you imagine the number? Okay, and they are a variety of shapes as we discussed. What are the variety of shapes that we see? Different kinds of shapes of bacteria. They are cocci. We will study all of them in detail. Bacilli and spirally. Okay, and these bacteria, they are important to many nutrient cycles. These bacteria are, they are important decomposers. Okay, I am writing here the heading also. Okay, so these bacteria, they play an important role as decomposers. Okay, what do? They decompose the organic material, and they are found all over the human body. Okay, so here only I'll write found all over the human body. Okay, so are they harmless or are they beneficial? Okay, are they harmless or are they beneficial? So majority of them are harmless. Okay, majority of them are harmless. That means they, some are beneficial also. Okay, and some bacteria are pathogenic. So majority are harmless and some are pathogenic. Pathogenic means causes diseases. Okay, so what is the history of bacteria? What is the history of bacteria? So I think you all know about it. What is the history of bacteria? So history of bacteria is, there is a scientist named Anton von Leeuwenhoek. Anton von Leeuwenhoek, this is U, Leeuwenhoek. He was a Dutchman. Okay, he was a Dutchman and he was the first person to see bacteria. Okay, he was the first person to see the bacteria. And he first observed bacteria through his single lens microscope. So what he did, I'll highlight some key points which led to the discovery of bacteria. So he was the first who observed bacteria through what? He was the first one who has observed bacteria through single lens microscope in the year, single lens microscope in the year in 1674. And he was the one who made his own simple lenses. Anton von Leeuwenhoek, he was the one who made his own simple lenses. Okay, he was the one who made his own simple lenses, and during his lifetime he made more than 250 microscopes. Okay, he was the one who made more than 250 microscopes. Can you imagine? Was made by him. Okay, What kind of microscope? Which consists of home ground lenses mounted in brass and silver plates. I'm not going into detail of all that. And his greatest discovery, Anton von Leeuwenhoek's, his greatest discovery was what? His greatest discovery was on June 16th. Is anybody's birthday on June 16th? No? Okay, fine. So on June 16th, 1675, it was his first greatest discovery when he saw bacteria. Along with bacteria, he saw fungi and many protozoa. Okay, and many protozoa in rainwater. And these organisms which he found, he called all of them what? He gave them a term. He called them animalcules. Animalcules. Please remember this term. Okay, and he sent a report of his findings of bacteria and algae to Royal Society of London. Okay, he sent his report to what report? This report. Okay, this report he has sent to Royal Society of London. And for this contribution, okay, he was given the title Father of Microbiology. So who is the father of microbiology? It is Anton von Leeuwenhoek. Okay, he was the one who was honored father of microbiology. Okay, so the term, so previously it was animalcules. Okay, then the term bacteria, which is a Greek word and which means small stuff. So the term bacteria, this bacteria is a Greek word, okay, which means what? Which means small stuff. It means small stuff, and it was first used in 1838. It was first used in, and by F.J. Kohn. Okay, and then comes another scientist named Louis Pasteur. What he did, he demonstrated that the fermentation process was caused by the growth of microorganisms or bacteria. So he was the one who demonstrated that the fermentation process was due to bacteria or microorganisms. Okay, it was his findings. And Pasteur discovered the process. Since the name Pasteur, he discovered what? He discovered pasteurization. What is pasteurization? Which is killing of bacteria by heating. So he discovered the term pasteurization, which is killing of bacteria by heating. Okay, and also he coined the term vaccine. Okay, he invented a number of vaccines. For example, he coined the term vaccine. And for example, the number of vaccine which was discovered by him was rabies. Okay, against rabies. And he also has studied the bacterium that causes the disease called foul cholera. Okay, so I'm not going into detail, I'm just writing he studied that he studied a bacterium that causes foul cholera. That causes foul cholera. Okay, so rabies vaccine was invented by him. He coined the term vaccine and all that. So this was about Louis Pasteur. Then comes Robert Koch. So we are still continuing with the history, the discovery of bacteria. Okay, so who was Robert Koch? He was a German doctor. He was a German doctor, and what he did, he showed the first time in 1876 that bacteria can cause disease. So in 1876, for the first time, he showed that bacteria can cause disease. Okay, that bacteria can cause the disease. And this was followed by Robert Koch's experiment. How he did, what was his next step? He has performed some experiments on bacteria as a source of a disease. Okay, he has performed some experiments on bacteria as a source of a disease, taking bacteria as a source of a disease. Which bacteria? Anthrax. Anthrax bacillus was his, he has taken that bacteria as a source of disease. Okay, he has performed several experiments on these, and for this he got a Nobel Prize in 1905 for this discovery that bacteria can be pathogenic. Okay, now another thing what he did, so this was his first, then second, then what third thing he did, he introduced staining techniques. You must have done the practicals related to gram staining. So he introduced staining techniques and also the methods of obtaining bacteria in pure culture. This we will learn when we will do applied microbiology. So methods of obtaining bacteria in pure culture. In pure culture using solid media. Okay, what he did, he discovered Bacillus tuberculosis. Okay, this Robert Koch, what he did, he discovered Bacillus tuberculosis, the bacteria responsible for causing TB, in 1882, and Vibrio cholera in 1883. So let's understand about the experiment performed by Robert Koch to prove that bacteria can be pathogenic. So first of all, what he did, so let's discuss about the experiment. Okay, so what is the first step he has taken? So first is microorganisms are isolated. Microorganisms like bacteria, they are isolated from dead animal, maybe mouse, isolated from dead animal. What is the next step? That there is a two-step following to number one is 2A and another is 2B. In 2A, what is done, that two kinds of experiments, there is a bifurcation after the step 1. In the first case, the microorganisms, these microorganisms which are isolated, these microorganisms are grown in pure culture. Okay, and the second case, the microorganisms are identified. Okay, and the microorganisms are then from both the cases, the microorganisms are then injected into the healthy animal. Okay, these microorganisms are then injected into a healthy animal. Okay, after that, after the injection into the healthy animal, what is the fourth step? The fourth step is the disease is reproduced, okay, in the second animal, that is the healthy animal, and microorganisms are isolated from this healthy animal. Okay, so microorganisms are isolated from healthy animal. Healthy animal is the second animal, not the infected one or dead one. Okay, then again two experiments are performed, 5A and 5B. In case of 5A, pathogenic microorganisms are grown in pure culture, same like that happened in 2A. So in pure culture, and in the second case here identified as in case 2. Okay, so when they are identified, they are found that here the diseased bacteria or microorganisms which are transferred into the healthy animal, this diseased, okay, this diseased bacteria or the bacteria causing disease is reproduced. So we will find that the same kind of bacteria which were causing the disease here, it will infect the healthy animal also, and the same kind of bacteria is again identified, which proves that the bacteria can be infectious or it can cause disease. So next comes another scientist, Paul Ehrlich. Okay, I'm sorry, it will not be Ehrlich. It will be Ehrlich. So anybody's Ehrlich fan here? Must be, right? He's such a nice singer. So okay. So Paul Ehrlich, he was the German medical scientist, developed the first theory concerning how bacteria cause disease. So bacteria cause disease was proved by Robert Koch, and he was the one who was concerned about how bacteria cause disease. Okay, how bacteria cause diseases and how the immune system fight with these microorganisms. Okay, such an analytical mind he had, right? How immune system fights these microorganisms. So then again comes another scientist, Wecker. He was a young Dutch pathologist. He was working on yellow disease of hyacinth. Okay, he was working on yellow disease of hyacinth. He was working on yellow disease of water hyacinth, and he proved that this disease is caused by bacteria. Okay, he proved that the causal organism is bacteria. And his work got published. Okay, his work got published in which year? In 1883, at the time when there was discovery about bacteria was going on. Okay, then comes another scientist named Dr. Elie Mechnikov. He was a Russian immunologist. Okay, he was a Russian immunologist, And in his early 1900s, he suggested that a synergistic interaction exists between bacteria and host. What he said? He said that there exists a synergistic relation or interaction exists between bacteria and host. And their host. And this process is known as phagocytosis. Okay, and there were few more scientists which we just need to know about, like Hansen, like Oxton, like Loeffler. So what did they do in 1874? Just try to remember the chronology. And 1881 and 1884. Okay, in 1874 he described what? He described leprosy bacillus. He described leprosy bacillus. Okay, Oxton did? He discovered staphylococcus. And what Loeffler did? He isolated diphtheria bacillus. Okay, so Hansen described leprosy bacillus, Oxton discovered staphylococcus, and Loeffler discovered what? He isolated diphtheria bacillus. So here we complete about the history of bacteria. Now we proceed with the evolution of bacteria. So in the second part we will continue with the bacteria. Here evolution of bacteria. So how did the evolution of bacteria took place? So these bacteria, they are thought according to some of the scientists to be the first forms of life. Okay, so according to some of the scientists, it was thought that they are the first form of life. Okay, which according to some scientists it was, and it is about 4 billion of years ago this thought was there. Okay, which is about 4 billion years ago. Then it is believed that both the domain archaea and the domain eukarya. What are these? These are two domains, archaea and eukarya. They have evolved from bacteria. Okay, and due to poor fossil record, scientists are unable to determine from where bacteria evolved. Okay, proper information was not there, and these bacteria then they can be autotroph or heterotroph. So these bacteria, they can be autotroph or they can be heterotrophs. Okay, they can be autotrophs or they can be heterotrophs, and the ones which are classified as autotrophs, they are photosynthetic or chemosynthetic. They are either photosynthetic or chemosynthetic. Photosynthetic are what? Photosynthetic, you already know that they can obtain the energy from sunlight, and chemosynthetic are the ones they get the energy by breaking down inorganic substance. Okay, by breaking down inorganic substance. Okay, for energy. So this is about the bacteria photosynthetic mode, and bacteria which are heterotrophs, they derive the energy from breaking down complex organic compounds. Okay, so they derive energy. How? They derive the energy from breaking down complex organic substance. Please remember, complex organic substance is from there they get their energy from. Okay, and where they get this organic compound from? They get it from the environment. And examples of heterotrophs, they include example saprophytic bacteria. Okay, saprophytic bacteria. And the saprophytic bacteria, they depend or they feed on decaying matter. Okay, they feed on decaying material and organic waste. Okay, and these can also be the one that live as parasites, and the ones which are parasites, these parasites, they depend on the living host. Okay, they absorb nutrients from living host. Okay, so saprophytic from dead host and parasitic from living host. Saprophytic from dead host. Okay, so so depending on the species, these bacteria, they can be either aerobic, which means they can require oxygen to live. So bacteria, they can be either aerobic are the ones which require oxygen to live. They require oxygen to live. And they can be anaerobic. Anaerobic means they do not require oxygen. Okay, don't require oxygen to live. Okay, and oxygen is deadly for them. They will not grow. Okay, so next we come to the prokaryote and eukaryote evolution. Okay, our next topic is prokaryote and eukaryote evolution. So starting with cellular evolution. In cellular evolution, what we need to know is that, so in cellular evolution, the current evidence indicates that eukaryotes have evolved from prokaryotes between 1 and 1.5 billion years ago. Okay, so cellular evolution says that eukaryotes have evolved from prokaryotes. Okay, and there are two theories regarding that. What are the two theories regarding that? One is infolding theory and another is endosymbiotic theory. One is infolding theory and another is endosymbiotic theory. Okay, so what is there in endosymbiotic theory and what is there in infolding theory? So in case of infolding theory, the infolding of the prokaryotic plasma membrane gave rise to eukaryote organelles. Okay, so if this is the genetic structure and if this is the plasma membrane, there is the infolding of the plasma membrane. Okay, and this infolding of the plasma membrane give rise to eukaryotic organelles. Okay, like this. This is the genetic material. Okay, so infolding of this is plasma membrane and this is organelle. Got it? Another theory is endosymbiotic theory. In endosymbiotic theory, what it says that, first of all, what is the meaning of the term endosymbiosis? So endosymbiosis, it refers to one species living within another. Okay, one species living within another. And what is the another species called as host? Okay, and movement of some smaller photosynthetic and heterotrophic prokaryotes into larger prokaryotic cells. Okay, say for example, movement of some smaller photosynthetic and heterotrophic prokaryotes. Some photosynthetic and some heterotrophic prokaryotes, okay, into movement into some larger prokaryotic host cells. So some small prokaryotes, when they move into larger prokaryotic host cells, they form cell organelles. So these form cells organelles in the larger prokaryotic cells. So here I draw. So if this is a larger prokaryotic cell, and if this is, say for example, mitochondria, if this is chloroplast, okay, this is cristae, this is granum, okay. So if this is mitochondria, MT, and this is chloroplast, CHL, I'm writing in abbreviated form, and this is the genetic material of the prokaryotic cell. So this, when takes entry into the larger prokaryotic cells, okay, it behaves as organelle. Okay, so this is now an organelle. Got it? So again, in the next part, what are the earliest prokaryotes? Okay, in the next part we would like to talk about the earliest prokaryotes. What are the earliest prokaryotes? So most numerous organisms are there on earth, okay, and the earliest fossil dates like 2.5 billion years old. It was found that the earliest bacteria, earliest prokaryotes, once they are said to be as bacteria, okay, and it was from the fossil records. And next we move to classification, okay, where we also have to study about the bacteria, where how bacteria is similar to the three other domains of life. Okay, so next we start with classification of life. So in classification for life, there are three domains of life. Okay, what are the three domains? What are the three domains? The first is archaea, the second is bacteria, and the third is eukarya. Okay, what does archaea include? Archaea are prokaryotes which lives in extreme environment. They are the prokaryotes which live in extreme environment. Bacteria like cyanobacteria and eubacteria, we will discuss about each of them in detail as we gradually move forward. Cyanobacteria and eubacteria. And eukarya includes what? Protozoans, fungi, plants, and all evolved life forms, animals, etc. Okay, Now coming to kingdoms of bacteria. Okay, classification of bacteria we also have to study in detail. It's there in your syllabus. Now moving to kingdoms of bacteria. What are the kingdoms of bacteria? Starting with first Archaeobacteria. So as the term archae is there, you will find that they are growing in extreme environment. So they grow in extreme environment. Okay, what are the extreme environment? Like undersea volcanic vents. Then what others are there? Acidic hot springs. Then what else? You can also write in the comment box if you have seen some extreme bacteria. The ones which are recently discovered will be like given appreciation. So try to give your inputs as well. Okay. So that will make our content more engaging. So sometimes there may be opportunity that I'll be taking your live classes also, right? So Archaeobacteria are the ones growing in the extreme environment. Then comes the eubacteria. Eubacteria are the ones. What are eubacteria? They are the true bacteria. They are the true bacteria, and most bacteria in this group are photosynthetic cyanobacteria. Most are photosynthetic cyanobacteria. Most are photosynthetic cyanobacteria. We will discuss about them. Okay, don't worry that ma'am, what is cyanobacteria, what are true bacteria? We will discuss about all of them. Then coming to characteristics of bacteria. Characteristics of bacteria. So what are the characteristics of bacteria? Already we have talked about some of their characteristics when we started with that they are microscopic. What else? There is no true nucleus. Okay, no nucleus. And if any kind of nucleus is present, primitive nucleus is present, then it is not membrane bound. Okay, and no membrane bound organelles. Okay, no membrane bound organelles are there. No true nucleus is there. They contain ribosomes for protein synthesis. Ribosomes are the organelles for protein synthesis. And single circular chromosome is there. Single circular chromosome. What is type of chromosome? Circular. Because in bacteria you will find circular chromosome is there. This might be asked in some competitive exams also. Okay, so single circular chromosome in nucleoid region. Sometimes it's nuclear, it's just primitive nucleus. Okay, so there is a protective layer in bacteria. How do they protect themselves? There is a protective layer in bacteria. Okay, their cell wall is made up of peptidoglycan. Cell wall made of peptidoglycan. And they have a sticky coating. Okay, they have a sticky coating. They have a sticky coating called capsule. Okay, they have a sticky coating called capsule. And this capsule, what is the role of this capsule? Capsule helps in attachment. It helps in attachment to host or other bacteria. Okay, it helps in attachment to host or other bacteria. What is the role of the capsule? Where, in which type of microorganisms are they present? So they are present in bacteria, and that helps in attachment of the host to other bacteria. Okay, and these capsules, they are sticky in nature, which helps in attachment. Okay, if this is the capsular structure, they are sticky in nature. Okay, microscopic view. Okay, now these bacterial structure, they have small rings of DNA called plasmids. Okay, I am continuing with the characteristics only. So these bacterial structure, they have small Rings of DNA, small rings of DNA. And what is this small rings of DNA called as? Called plasmids. These bacteria, they are microscopic, they are also unicellular, and they are very small in size. What is the size? 0.5 to 2 micrometer, so small. Okay, and these plasmids, they are what? They are ring-like structure. They are ring-like structure. Okay, helical. So moving forward, that the enfoldings which are there, the cell membrane enfoldings, okay, the cell membrane enfoldings of this bacteria, they have a specific name. Cell membrane enfoldings, they carry on photosynthesis as well as cellular respiration. And cellular respiration, what are they called as? What are they called as? They are called as, these enfoldings are called as mesosomes. Mesosomes. Okay, very important. What are mesosomes? There is an enfolding onto the cell membrane, and this carry out photosynthesis as well as cellular respiration. Okay, now these bacteria, they grow at pH 6.5 to 7, which is nearly neutral pH. Okay, and some act as many act as decomposers. So I write here, many bacteria act as decomposers. Some are pathogenic. Some are pathogenic as well. Okay, pathogenic means cause disease. Next, coming to flagella. So these bacteria, they are motile and they have flagella. So these are again into characteristics of bacteria. So these bacteria, they have flagella, which makes these bacteria motile. So they are motile appendages which helps bacteria to move. Okay, and these flagella, they are attached by basal body. Okay, they are attached by basal body. We'll study about the structure of flagella later on. And a bacteria can have one or many flagella. Okay, there can be one or many flagella. This is monotrichous, one flagella. Cluster of flagella from one point, then flagella from both sides, biflagellate, then the cluster of flagella originating from anywhere. Okay, so you have to write the names of all that in the comment box. Got it? You have to write down the names of all of these flagella in the comment box. Okay, so next is flagella. This flagella is made up of flagellin, made up of a protein named flagellin. Okay, and this is used for classification. Monotrichous is one flagella, monotrichous. Okay, so I am making your work easier. Now you do not have to write in the comment box. You have to just learn and you have to be very thorough with the topic. Okay, one flagella. I'm just writing a number of flagella in bracket 1. And lophotrichous, where the tuft coming out from one place, this is lophotrichous. The tuft coming out from one place, lophotrichous, which is the tuft coming out from one place, tuft of flagella. Okay, then is amphitrichous. Amphitrichous, when the tuft is at both ends, amphitrichous. both ends. Then it is petritricus, peritricus, peritricus, which is all around the bacteria. Okay, so when a tuft is from one end, it is lophotricus. When it is all over the body of the bacteria, it is peritricus. So this is about flagella. Next we come to pili. So next we come to pili. So what are these pili? They are short protein appendages. That was for flagella was for the motility of the bacteria. But what was the purpose of pili? So pili are shorter than flagella. Okay, so they are short protein appendages. Flagellin protein was also proteinaceous in nature. So they are short protein appendages and they are smaller than flagella. They are smaller than flagella and they adhere bacteria to surface. What is their role? They helps in adherence of bacteria to any surface. Then they also help in conjugation. Okay, they also help in conjugation for exchange of genetic material. For exchange of genetic material. And they aid flotation by increasing buoyancy. They aid flotation by increasing buoyancy, making the bacteria light. Okay. BUOYANCY. Okay, so this pili are mainly for conjugation. They make the conjugation tube and helps in transfer of genetic information, which we will study in the later part of this class. Then comes the shapes, which are used to classify the bacteria. Okay, next is bacterial shapes. So number one is bacillus. So bacillus are generally what kind of shape? They are rod shaped. Next is coccus. COCCUS. Coccus are generally spherical in shape. Spherical means round in shape. Okay, next is vibrio. So vibrio, they are comma shaped and they have flagella. And the next is spirillum, which is spiral shaped as the name says. Next is spirochaete. They are worm-like and they are also spiral. Okay, they are worm-like and they are also spiral. Next we move forward to, I must draw the shapes as well. So the rod shaped, what is the rod shape look like? They are rod, simply rod shaped. This is spherical shaped, okay, or round shape. Example, coccobacillus. I'm not writing the example right now. You have to find out the examples by yourself, okay? So next we come to you have to find out the examples by yourself. So vibrio, they are kind of comma shaped. Next is spiral shaped, which is spirillum. Spirochaete, they are more of a spiral and a kind of worm. Okay, so they also have vibrio also have flagella on it. Okay. So next, moving to grouping of bacteria. Grouping of bacteria. So if they are 2 in number, it is diplo. If they are chains, they are strepto. So this terminology as a prefix, they are used when they are into a specific groups. So diplo when they are 2 in number, strepto when they are in chains, and staphylo when they are grape-like clusters. Okay, so this is diplo when they are 2 in number together, diplococcus, example diplococcus. When they are in chains, it is streptococcus, and since they are spherical, so I should write coccus. Then when they are into clusters like grape, it is staphylococcus. Okay, so interesting to know, right? So this streptococcus, which are they in chains, this streptococcus, they cause a specific kind of disease. What they cause? They cause strep throat. They cause strep throat. Okay, it's a disease. Then comes the staphylococcus. Staphylococcus when they are in chains. Then there are streptobacilli also. Bacillus are generally rod shaped. So I write here, it can be streptococcus, it can be streptobacilli. Okay, depending upon the shape. Okay, spirillum is the spiral shaped, then spirochaete are worm like spirals, and then coming to bacterial kingdoms. So before that, before moving forward, I would like to give you an assignment or self-test. So let's start. So can you tell me what shape of this bacteria this is? Quickly write it in your copy and then we will discuss the answer. I am just drawing the shape, you please write the answer. And then when I will discuss the answer, you please match whether you are correct or not. Number 1, number 2, number 3, number 4, number 5, number 6, number 7. So this is, the first one is what? The first one is rod shaped and this is bacilli or bacillus. The second one is coccus, spherical shaped. 2 in number, diplococcus. Many in numbers, in chain, streptococcus. Grape like staphylococcus. Comma like vibrio. And spiral is spirillum. Isn't it interesting? Next, coming to bacterial kingdoms. Bacterial kingdoms. In bacterial kingdoms, number 1, starting with archaebacteria. Starting with archaebacteria. AE, archaebacteria. So as discussed, they live in harsh environment. Okay, they live in harsh environment and they lack peptidoglycan. There has to be a different cell wall material which will allow them to live in the harsh environment. So peptidoglycan will not let them live in the harsh environment. They have a different kind of lipid in their cell membrane. So lac peptidoglycan have different lipids are present in cell membrane. Also many different kind of proteins are required for their function. So different kinds of ribosomes are present. Different types of ribosomes are present. Different heat shock proteins and all that are required for their function. And their gene sequence is also different. Okay, so these archaebacteria, the most important point is that they live in harsh environment. They live in harsh environment. I'm sorry, the content got deleted, so I had to write it again in a different slide. So you will find there is a little difference from the last slide and this slide. So I continue. They live in harsh environment and they do not require oxygen. They do not require oxygen to live. Okay, they can live in extreme weather conditions and they are called as ancient bacteria. Okay, they are called as ancient bacteria. And these archaebacteria, they are subdivided into three groups. What are these three groups? Number one is methanogens. Number two is thermoacidophiles. And number three is extreme halophiles. So let's start with methanogens. So methanogens, they live in anaerobic environment. They live in anaerobic environment means devoid of oxygen. Anaerobic environment, that is environment without any oxygen. So how do they get their energy from? They get energy by changing H2 and CO2 into methane gas. So they get energy by changing H2 and CO2 into methane gas. They get their energy by changing H2 and CO2 into methane gas, and they are found in swampy areas. They are found in swamps, sewage treatment plants, digestive tract of animals, etc. What do they do? These methanogens, they break down cellulose in animal stomach. What they do? They breaks down cellulose into what? And produce methane gas. Okay, this breaks down cellulose in cow's stomach to produce biogas. Okay, which is methane, and they produce methane gas. Okay. So next are thermoacidophiles. Number two was thermoacidophiles. So these thermoacidophiles, they live in extreme hot condition. They live in extremely hot environment and they are found in volcanic vents. Hot springs, okay, hot springs and cracks on the ocean floor. that leak acid. And the last one, which are extreme halophiles. What are extreme halophiles? They live in very salty condition. They live in very salty weather, salty water, and use salt to generate ATP. Okay, they use salt to generate ATP. And what is ATP here? It's energy, and they are mainly found in Dead Sea, Great Salt Lake inhabitants, and other places as well. You can give your input in the comment box. So this was about the kingdom Archaebacteria. Second, so I write here, first is Archaebacteria. Second is Eukarya. Okay, what is third? What are third? Is there any? Is there any? The third is Cyanobacteria. Okay. So starting with the kingdom Eukarya. I am sorry, it will not be Eukarya, it will be Eubacteria. My mistake, I mistook it with the three domains. It will be Eubacteria. Okay, so don't get confused with the domain and the kingdom. Eubacteria. Okay, that was Archaea, Eukarya, and what was the third one? But here it is Archaebacteria, Eubacteria, and Cyanobacteria. Okay, there it was Archaea, bacteria, and Eukarya. I write here so that you will not be confused. Three domains. What are the three domains? Three domains are Archaea, which is not Archaebacteria. Okay, then bacteria itself, then it is Eukarya. Under Eukarya, higher plants, higher complex organisms are there. Inside bacteria, it is Archaebacteria, it is Eubacteria, and it is Cyanobacteria. I hope you understand, right? So now we will start with Eubacteria. Second kingdom, Eubacteria. So Eubacteria are the true bacteria. Okay, what are the characteristics? That there are three basic shapes. Three basic shapes. What are the three basic shapes of Eukarya? They are coccus, bacillus, and spirilla. And most of them are heterotrophic. Most are heterotrophic. That means they cannot make their own food. They may be aerobic or anaerobic. They may be aerobic or anaerobic. And they are identified by gram staining. What is gram staining? We will discuss. They are identified by gram staining. So what is gram staining? So it was not related to gram anywhere, just because the scientist named was Gram, hence the name is gram staining. So it was developed in 1884 by Hans Gram. Okay, you must be wondering why it is called gram staining. Does the gram has to is there any connection with the gram or not? Okay, so no connection, just because of the scientists, it's hence named gram staining. And the bacteria treated with purple crystal violet stain. Here a stain is used named crystal violet stain and saffronin stain. Okay, and the cell walls, if they are stained with crystal violet, it gives purple color, and if they are stained with saffronin, it gives reddish pink color. Okay, so which kind of cells take which color? Okay, so depending upon the stain which they take, there are gram positive and gram negative bacteria. So gram positive bacteria and gram negative bacteria, depending upon the stains which they take. Okay, gram negative bacteria. So starting first with gram positive bacteria. Starting with gram positive bacteria. So gram positive bacteria, they have thick layer of peptidoglycan. They have thick layer of peptidoglycan. And what is peptidoglycan? We will discuss it. It is protein sugar complex. It is protein sugar complex. Okay, and in case of gram positive bacteria, it is a single lipid layer. They have a single lipid layer. They take the stain and become purple in color. Which stain? Crystal violet stain, and purple in color. Crystal violet stain. And they are stained purple with crystal violet, and they can be treated with antibiotics. When antibiotic is given, they digest the cell wall of this gram positive bacteria and they dies. So they can be treated with antibiotics. So example of gram positive bacteria, they include what? They include lactobacilli. What does lactobacilli do? It makes yogurt. Buttermilk, etc. Then actinomycetes. Actinomycetes, they make antibiotics. Then it is Clostridium. Clostridium, they locks the jaw. Sometimes we see that the person's lower jaw and the upper jaw becomes locked. It's a very serious disease. A person faints. Okay. So tetanus is also given at that time in some cases. Then it is Streptococcus. They strep the throat. Then it is Staphylococcus. They all are gram positive. Staphylococcus, what do? They staph infections. Okay, now next, gram negative bacteria. Gram negative bacteria, what do? What is their structure? How do they stain? So they have a thin layer of peptidoglycan. Okay, they have a thin layer of peptidoglycan. Okay, and the lipid layer is extra thick. So extra thick lipids. Okay, next is they stain pink with saffronin. Pink or reddish pink with saffronin stain. Okay, and they are hard to treat with antibiotics. They are hard to treat with antibiotics because they have such a thick lipid layer. Okay, so in case of gram positive, we saw that they are like lactobacillus, actinomycetes, and all that. Those were example. And here in case of gram negative, they some are photosynthetic, but make sulfur, not oxygen. Okay, they make sulfur, very important to know, but not oxygen. Some also fix nitrogen for plants. Okay, because plants cannot fix their own nitrogen, it takes the help of bacteria. Okay, so example, rhizobacteria. Rhizobacteria, they live in symbiosis with the nitrogenous plants like legumes. So they grow in root nodules. These rhizobia, they grow in root nodules. Of which plant? Of leguminous plant. Leguminous plants, like example, Fabaceae family of plants, like dal and all that, like soybeans, pea, etc. Okay, so the roots, due to the presence of the bacteria, the nodules are formed in the roots, and inside these nodules the bacteria fix the nitrogen. Bacteria live inside this root nodules, okay, and fix nitrogen there into these nodules. Okay, they fix the nitrogen into ammonia. These bacteria fix nitrogen into usable ammonia, so that this ammonia can be incorporated into amino acid, incorporated or used up to form amino acids. And what is the amino acid used for? These amino acid will help in the formation of proteins. Clear now? So what kind of diseases are caused by these gram negative bacteria? These gram negative bacteria, example, Rickettsia. They are parasitic. They are parasitic bacteria which are carried by ticks, and this cause Lyme disease. This cause Lyme disease and Rocky Mountain spotted fever. Okay, this is a gram negative bacteria, Rickettsia. So this is A and then E. Okay, I write it properly so that it will be helpful for you if you want to write the notes. Okay, this is A. Next is coming to the third kingdom of bacteria. It is cyanobacteria. Third kingdom of bacteria, cyanobacteria. Okay, so these cyanobacteria, they are gram negative. Okay, these cyanobacteria, they are gram negative and they are photosynthetic. Okay, these are photosynthetic, gram negative, and they are blue green. They are blue green bacteria. I am sorry, here this kingdom of bacteria. Okay, so I am omitting it here, so I think I should remove it from there as well. So consider two kingdoms. There are two kingdoms, archaebacteria and eubacteria. Okay, cyanobacteria won't be there. Okay, they are just group of bacteria and cyanobacteria won't be there. So I have cut it out from here also. Bacterial kingdoms are archaebacteria and eubacteria. Please don't write there cyanobacteria, right? So then moving forward, we are into cyanobacteria. So these cyanobacteria, they contain a pigment known as phycocyanin. They contain a pigment known as phycocyanin, which is red-blue pigment. Okay, and also contain chlorophyll. Phycocyanin and chlorophyll. And these cyanobacteria can be blue-green, they can be red, they can be yellow, they can be of several colors, right? Yellow, brown, black and blue-green have already been written. Then they may grow in chains. Okay, if they grow in chains, example include oscillatoria. Please remember the examples. Okay, they are very important. And these cyanobacteria have a very important structure known as heterocyst. What is the role of heterocyst in a cell? That these heterocysts, they help in nitrogen fixation. So inside themselves they can fix the nitrogen due to the presence of heterocyst. Okay, so some cause eutrophication. Some of the cyanobacteria, they cause eutrophication, algal bloom. Okay, so some cause eutrophication. What is eutrophication? That they use oxygen, okay, when die and they decompose in water. So eutrophication term we may discuss elaborately later on. Then comes another type of bacteria, which is spirochete. Okay, so spirochete we just need to know about. So this cyanobacteria is also gram negative. So another gram negative bacteria we need to know about is enteric bacteria. Enteric in intestine. Okay, so they can live in both aerobic and anaerobic habitat. So both aerobic and anaerobic. Both anaerobic and aerobic environment in which they can live. Example E. coli, which can live in intestine. Example Salmonella, which cause food poisoning. Okay, and this enteric bacteria, after that there is another bacteria which we need to know about, a gram positive bacteria, which is spirochete. Okay, so let's go back to where I have written spirochete. They are worm like spiral. Okay, the structure we have learned, they are worm like spiral shaped when we read about the classification of bacteria according to the shape. So they are gram positive and they have flagella at each end. What is that called as? When the flagella are present at each end, what is the term you call it as? Please recall. It is, if they have two flagella, it is biflagellate. But when flagella is there at both ends, what you will say that this as? Please tell me. It is Amphitrachus. Okay, so and these kind of bacteria, spirochete, they move in corkscrew motion. Corkscrew, how we move the corkscrew, in that way they move. And some are aerobic or some are anaerobic. They can be aerobic or anaerobic. I'm just writing in this way. And they may be free living, they may be symbiotic, and they may be parasitic. Okay, then coming to another kind of bacteria which are chemoautotrophs. So first is autotroph, then is heterotroph, then it is chemoautotroph. Chemoautotrophs are kind of gram-negative bacteria again. So gram-negative bacteria again, this is chemoautotroph. Chemoautotrophs are gram-negative bacteria and they obtain the energy from minerals. They obtain energy from minerals like iron. And they are found in freshwater ponds. They are found in freshwater ponds. Okay, so next we will start with in detail about the peptidoglycan, what is the chemical nature of peptidoglycan, how it is made up of, what is the cell wall composition in gram-negative bacteria and gram-positive bacteria, how it is helpful for the survival of those bacteria. So this we will continue.